TF Sites is an all-in-one toolkit for enhancer visualization and analysis for researchers from all backgrounds. Enhancers are sequences that determine when and where genes are expressed. Combinations of transcription factors bind to binding sites within the enhancer to activate tissue and time-specific expression of genes. Sequence changes that alter transcription factor binding sites can drive changes in gene expression, most commonly through increasing the affinity of the site or creating de novo sites. These sequence changes can have important implications in disease and evolution applications. We therefore design TF sites to both visualize predicted binding sites and their affinities within enhancers, as well as report which sequence variations alter binding of transcription factors. Although we developed the tool with interests in studying enhancers, this tool can also be applied to study promoters as well. TF sites contains 3 tools: visualize TF sites on sequences, find TF sites altered by sequence variation, and compare TF sites across sequences. The first tool, visualize TF sites on sequences, predicts and visualizes binding sites within enhancers. It can assign each binding site a score or affinity value depending on the type of reference data given. For example, we are interested in studying the ZRS enhancer, which we have shown a region of below. This enhancer regulates expression of the sonic hedgehog gene in the developing limb of vertebrates and is heavily implicated in developmental defects. We are interested in annotating transcription factor binding sites on the ZRS enhancer, and we can use TF sites to integrate different types of reference datasets to do this. The first type of data that can be used is PWM data, which is publicly available on databases such as Jasper and Hokomoko, and has been the historically popular method of scoring binding sites. While PWMs have been extremely helpful in understanding predicted binding sites, it is known that they have a difficult time detecting low affinity sites, and their binding score does not correlate well with relative or absolute affinities, likely due to an assumption that each nucleotide contributes independently to binding strength. Therefore, when available, relative and absolute affinity datasets are preferable. One common example of relative affinity data is from protein binding microarrays, or PBM. PBM data is a useful technique to assess binding sites because it is an empirical method of measuring the binding between a transcription factor and DNA sequence. This data is accessible on the UniProbe database. This type of data can be used to assign each binding site with a value called a relative affinity. PBM correlates with methods that measure absolute binding affinities, which is also our last category of data, and includes techniques such as mitomy or BET-seq. We host both PWM and PBM relative affinity data on the TF Sites website itself. For example, we can use this tool to annotate ETS and PBX transcription factor binding sites on the ZRS enhancer. So for ETS, we can use mouse PBM relative affinity data, and for PBX, we can use position weight matrix or PWM data to score the binding sites. And this is what our analysis would look like. Each transcription factor is annotated on the ZRS enhancer, and it's labeled with the name of the transcription factor, its position, a unique ID associated with it, its orientation, and its assigned affinity or score. The second tool in TF sites is find TF sites altered by sequence variation, and this predicts how every possible single nucleotide substitution affects TF DNA binding. Mutations within existing binding sites can increase or optimize affinity, reduce the affinity, or ablate the site completely. Mutations outside of existing binding sites can create de novo binding sites. For example, we can use the ZRS enhancer again to look at saturated mutagenesis for ETS. We can use the mouse PBM affinity data as we did before, although we could also use PWM data or other types of affinity data as well. And here's what our analysis would look like. Blue represents changes in nucleotides that increase the affinity or score of the binding site. Orange represents decreases. Green is for created binding sites, and red is for deleted binding sites. The intensity of the color is determined by the assigned affinity or score of the site. The third tool is compare TF sites across sequences, and this tool reports binding sites that are altered across 2 or more enhancer sequence variants. In biomedical applications, this tool can analyze reference and alternate alleles that are associated with diseases or changes in gene expression. In evolutionary applications, this tool can be used to determine which binding sites are lost, gained, or changed within a particular clade of species. For example, we can look at changes in S transcription factor binding sites between these 2 sequences using affinity data. Comparing the original and mutated sequences, one ETS binding site reduces in affinity. And here we can see the binding sites in both sequences as well as how they compare to the binding site definition. We also have a binding site that is created, meaning it wasn't in the original sequence but now exists in the mutated sequence. Similarly, we can perform another analysis instead using PWM data to search for changes in LHX binding sites. Instead of scoring the sites using a binding site definition, we use the motif. So here we have an example of a site that is optimized, so its score is increased, and another where the site is deleted. In whole, TF Sites empowers researchers from diverse backgrounds to reveal binding sites within enhancers and generate hypotheses of the mechanisms behind enhancers relating to disease, development, and evolution. Thanks for using TF Sites and good luck with your research.